Bio-Nano things sounds a bit spooky, but I have to share one distant memory we had ages ago. I think it was rather the late 90s or the beginning of the 2000s. There was a paper and presentation of Stefan and a mutual friend, and they talked about mobile networks, which was fancy at that time, and then they said, well, if you had small helicopters that could, today we would say drones or something like that, you could try to detect gaps between the network parts or partitions and try to make a cover there and close the whole thing. Everybody in the room was sitting there finding reasons why this was a stupid idea. Remember Otto Spaniol and others and making fun of it. And something like 10, 15 years later you say, oh actually, we did a lot of injustice to that. It was such a brilliant and early idea. I think this is one of the reasons, among many other reasons, why it's worth listening to what Stefan Fischer has to say. And so I'm happy to introduce Stefan Fischer to you, professor at the University of Lübeck, if I remember correctly, and a dear friend.
Thank you very much, Christian. Yeah, good point. Actually, I'm still a bit unhappy that we didn't follow our work on drones and so on. I think we by now we would be world-leading researchers in this field. So, second time this should not happen to us. So I'm going to talk about Internet of Bio-Nano things, and actually, just to, if you want to summarize this in one word, it's artificial communication within the human body. That's the idea. How could you do that? And what I'll do is I'll first present, let's say, the basics of something we would call it nanorobotics, nanocommunication, and this also is let's say, a different part. When we started working on this, which is already more than 10 years ago, we always used the term nanocommunications. Meanwhile, no one is using that anymore, like wireless sensor networks. Usually we say Internet of Things. What people say now is molecular communication, because it's not really the same, but it's the way of communication within the body will quite certainly work in the future. And this is let's say I'll talk about this most of this talk here. As you can imagine, we are also looking at one major application, and the application we are interested in is medicine, precision medicine to be precise, quite obviously, I mean, what would be the reason to communicate within the body? Certainly to run some kind of application within the body. I can't really think of any other reason to some kind of medical application. You know? And for that reason, I'll also briefly talk about this, give you an idea of what even medical people think nanotechnology could do within the body, and especially the nanotechnology that we are talking about here. Just like Christian, after this general introduction, I'll also do a famous presentation of our own work, the DNA-based nanonetworks part here. So we believe that this is one rather promising technology which could be successful for creating the Internet of Bio-Nano things. And for the rest, we'll see how much time is left. Maybe I'll talk a bit about gateway technology. So just imagine if you want to, if your nano network finds something within your body, or if you want to instruct the nano network to do something, you have to somehow communicate with it. So you need some kind of gateway. You need to be able to transfer information or commands from outside to the inside of the body, and the other way around. And there's also some interesting ideas and technologies of how you could do this. So, to be honest, what I'll show you here in the first part, that's mostly concepts, and even rather theoretical concepts, even in terms of computer science. But we are also working on and thinking in the direction of realizing the whole thing. And for the first time in my life, really, I stopped studying biology in 10th grade of school, and now I'm back in it. So we think about doing wet lab experiments and in vivo experiments. So something completely new, and probably also something very new to most computer scientists. Maybe some application, medical application examples, and then I think we are done. This guy here, by the way, is just to introduce, that's me, more or less. I asked DALL-E a year ago, so when I was 57, I asked him to produce an image of a computer science professor, 57 years old, light hair, and round glasses. So I like this image a lot. And why do I say this? Because I'll show you a number of images that DALL-E has produced when I asked him to ask her to give me an impression of what she thinks what a nano network or a nano robot and so on is. And while this is looks rather visionary, it's also interesting to look at that, and I'll come to that when the topic is coming closer. Okay, first thing, nano. I mean, most of us here are somehow computer scientists, I guess, and our level is the micro level, yeah? Microcomputers and so on. So nano is typically not what we think about, and I don't want to discuss everything here, but interesting. So nano is more or less this level here, yeah? And this is, as you can imagine, this is an important biological structure for us because, as you've already seen, our nano network technology is DNA-based, and such a DNA structure here is in the range of 10 nanometers. Yeah? So, just to keep this in mind, that this is somehow the thing that we are talking about when we talk about nano robots, nano communication, and so on. Yeah? So if you want to build something out of DNA, then you have you have a clear picture of, or a clear idea of how big these things would be, and this is especially interesting when we look at the images that DALL-E has produced. Okay. So when we started with the whole thing, we first did something like a taxonomy. Again, I don't want to go into everything here, because at that time there were lots of, so it was like the Klondike gold rush at the beginning. Everyone started with ideas and concepts and so on, and we try to unite these in this taxonomy here, more or less. So there's let's say rather not very powerful objects, so nano object, nano structure, nano device, and then the most powerful object, certainly something like a nano robot, which has some kind of possibilities to sense and to act and so on. And then we also have this stuff here, nanoparticles, nanoclusters. There's probably you've heard about these. There's a whole area of research for something like magnetic particle imaging and so on. So you have magnetic particles, also nano-sized, which you can then use to do medical imaging, and which you can also use to be steered through the body by applying magnetic force from the outside. This is what we are not interested in. This is just passive. Yeah? We are interested in active particles, which in principle are able to communicate, in principle are able to do something actively. Also, not interesting for here, also, this is an early image that several people came up with, how a nanobot looks like, and if you look at that, this looks more or less like an IoT device.
Smart dust. Remember smart dust?
Smart dust, yeah, exactly.
Pretty much same thing.
Yeah, exactly, smart dust. Well, it turned out, at least we believe that it turned out, that obviously it won't work like that. Yeah, so making things just smaller by 1,000 or so is not really the solution. Yeah, so obviously we need something else, some other kind of solution. So first image, let's assume we have nanobots like that. Then it immediately becomes interesting to also let them communicate, build networks out of these nanobots. Why? Because they are typically, I mean, if they are nano-sized, they are very, very small and will be extremely difficult to put very much functionality on them. And resources like let's say, a CPU or memory and so on, will be hard to bring this on very small devices. So if you want to achieve some functionality which can be set up in the human body, then you probably need something like a network or a distributed system of nanobots. So we need some kind of communication or at least information exchange capabilities. And this, I mean, the image here, if you think about nano DNA and so on, and you look at these fine technical structures here, it's quite obviously that it can't be like that. Yeah. So maybe if we ask Dali today or in 5 years or so, there might be a different picture coming up. But obviously, this is not the way of how these devices can look like. And well, probably most medical people would say if they see. If they use the, I don't know the English word, but all the Germans here know the Beipackzettel of the nanomedicine, and they see these devices here, they would probably, let's say, hesitate to use them. So if you have a nano network, then the next step obviously would be, as I already said, we have to integrate these nano networks into overall medical systems. Like body area networks. Yeah? If you have, let's say, if you have little nano networks here and here and here, and some devices attached to the body which communicate the usual way as body area networks communicate, then it makes a lot of sense to let them communicate to each other. Let's say to let the nano networks here communicate with the gateway, which is then able to communicate the information to, let's say, a medical device, like a monitor or whatever you could think of. That was an image that we, Falco Dressler and I, created in 2014, 2015. And today, that's an image that we used in a BNBF project proposal a year ago or 1.5 years ago.
Oh,
That's it.
almost fast. Welcome to Spain.
This concludes the session.
Kein Strom mehr. Die Kamera läuft. Die Kamera läuft. Ja, so ist das. Wieder komplett. Das hat man aber noch mit. Das Wi-Fi nicht geht, das kennen wir schon. Projekte nicht. Aber es
Ja, ich bin auch wieder aufgewacht. Danke.
Oh, das WLAN ist auch weg.
Da kommt was. Das ist schon wirklich. Das ist der Strom. Die Sache, da steht es nur so weit.
The beamer doesn't work.
Yeah, but the
Jemand hat versucht, beim Stromanschluss fängt an zu schließen.
Ja, freilich, was du da
Das ist der andere. Das ist keine Verbindung mehr. Das Mikro hat die Verbindung zum Sender, aber der Sender hat an-aus geht wieder.
Ja.
Ja,
Video.
Let's try with one. So we try without microphone. I think it works. Okay, so this is an image that we used for a BMBF proposal just last year or the year before. And it's basically the same image still, so the idea is the same. We have, let's say, the classical internet part here, and then the gateway, and here the internal body network. And you already see here, now we use the term molecular communication mostly. So communication based on molecules and no longer on radio waves. That's the main difference. And actually, when I say this, probably there's a lot of researchers in this area who would object now, because there's also a whole body of research work for radio-based communication, usually based on terahertz communication, because the antennas have to be so short that the frequency simply is very high. But I would say most researchers today believe that molecular communication is the way to go, and I'll continue with that topic now. Okay, so what do medical people think? What could be solutions? What could be applications when you use nanobots and nanonetworks within the human body? If you look up what medicine is, well, it's basically prevention, diagnosis, therapy, and rehabilitation. I leave out rehabilitation. I still don't have a good idea of what we could use that for. But let's look at the other three. What could you use that for? And now we again have the Dali images. So what is the guy doing here? He's removing plaque from the blood vessels. So plaque, as most of you know, or let's say the narrowing of blood vessels is one of the major causes of heart diseases and heart problems and so on. So if you have something like that, if you have devices which are able to patrol the blood vessels and remove all the plaque that is there, that could be very helpful. So prevention. And again, certainly, I don't say that with all the images now, but obviously this is not the way of how it will be. But let's say that this already looks a bit more realistic. So these are some scouts which check the blood vessels for tumors, for instance, or normal blood. And here we have a tumor, and then can act somehow if they once they find this. And this is medical people say this is one of the most important applications because finding issues like that, like a tumor cell or more tumor cells or any kind of illnesses, the earlier you find it, the better. So if you detect these kind of illnesses before any symptoms come up, it will be much better than if you detect them late. And then once you detect it, then you might have a nano laser, something like that, which is able to heat the whole thing up and simply kill it without affecting too much of the area. Okay, now not very realistic, but in terms of applications, this is what most researchers today believe is possible, not in this way, but in some other. So to be a bit more realistic, if we look at typical applications that we have in mind, that would be something like health monitoring. So we have a physician, a patient, and a nano network. And the basic idea is you have the nano network, let's say in some kind of tube, in a whatever, and you put it in your syringe, and then inject it into the patient, and then well, the patient will live with it. If you just look at health monitoring, so the nano network is doing its monitoring job, and usually everything is okay. But if something is coming up, like we have whatever situation here, the physician will be informed. The guy here is informed that he should quickly go to the hospital, whatever you could think of, or we could even have an intervention here by the nano network knowing by itself what it has to do, because it has been programmed for that before. or it might even be told to act in some way, whatever that some way would be. Okay, that looks much more realistic, and actually, most of the technologies that you see here are already available. They are not, let's say, they have not been used so far as a whole and in this way here, but everything you see here as single parts, so for instance here, nanostructures that you can inject is already all possible. Yeah? So it's no longer visionary what we talk about here. It's becoming more and more reality, apart from ethical issues and so on, but technically, and I'm just talking technically today, most of that is already possible. So that's the let's say that's the playing ground, and the question now is how do we realize that? How do we make that become reality? And the first question that we should ask is what are the requirements for a solution? And the first thing, and quite obviously, and probably the most important one, it has to be biocompatible. So if you have something, if you inject something in a person, and this let's say affects the immune system in a way that it goes berserk, then that doesn't help very much. Also the other way around, if the immune system simply kills all the devices that you inject, this also doesn't help very much. So biocompatibility is extremely important. Then obviously very small size of the devices, so then we have, as I said, this already very little functionality, so we definitely need some kind of distributed system solution. And we also need a solution for the construction on the nano level. And this is also something we have to think about. How do we construct, let's say, a nano robot in DNA size? Yeah? How do we do that? We don't know that yet, but I'll show you an idea of how this could be done. So that's there are certainly more requirements, but we think these are the most important ones. Okay, so let's talk about the solution that we believe could work. That's DNA-based nanonetworks. And the basics for that are DNA tiles. Has someone heard of DNA tiles already? This is a technology, let's say this is a concept which has already been developed, I think something like 20 years ago, by a guy called Eric Winfrey. He developed this in his PhD thesis. He's still a researcher in Pasadena. And he developed this idea to build, let's say, something like small building blocks which you can use to build more complex structures out of DNA. Why DNA? Because DNA is used in biology anyway as a building material. So it's very useful for that, and we show how we can use that here. So DNA tiles. So it's a bit similar to Lego bricks, but if you think about Lego bricks, how does that work? In principle, with Lego bricks, if you buy a set of Lego bricks, then you typically have a building plan, which you then use to, let's say, build this little robot here. Also. But I mean, you can also, using your fantasy, you can build whatever you like with Lego bricks. So actually, this is not exactly what we want. Yeah? So it's a bit similar. So we have different, let's say, different kinds of pieces, of building pieces that have different functionality and that we can assemble and that we can bring together, which then build huger structures. So it's somehow similar, but in the stuff that we want to do, we want to be sure that we only create a very special type of structure. Yeah? We have a clear idea of what kind of structure we want to achieve. And the question now is, do we have to do that ourselves, or is this happening or is this happening in a slightly different way? This is the important part here. Yeah? Self-assembly. What we think is an excellent solution for the building process problem. So imagine humans or machines or whatever trying to create small devices. This probably won't work, but the much better solution is self-assembly, because this is exactly how nature does it itself. Yeah? Molecules are built from atoms from other molecules and create bigger structures, and if we are able to control this process of self-assembly, so if we can instruct, let's say, the structures, how they come together and how they build new structures, then we are in a very good position. And this is exactly what we want to do. But now think about, let's say, putting all these Lego pieces here in one, let's say, in a box, shake the box, and then expect afterwards that the robot has come up. This is self-assembly. Yeah? Probably this won't work. It's a bit more likely with this here. Yeah? So if you are lucky, then some of these puzzle pieces which match would stick together. If you do this shaking process, still very unlikely. But in nature, this works actually much better than with our artificial stuff here, and we make use of that in DNA tiles. Okay. So Winfrey came up with a mathematical model and a biological equivalent, which is let's say extremely important if you want to come from concepts to real implementations. And the mathematical model of such a tile is first just a square. Yeah, it's called a block, and the block has glues on each side, and you can define how many glues you want to put on each side. And the glues have labels, and the thing can also have a marker in the middle. Yeah? So this is a typical tile, and the idea now is that you can connect these tiles to each other if they fulfill certain properties. So if you have another tile which also has two glues here and the label C here, then these two tiles would fit together and would click into each other more or less. Yeah, that's the idea here. And also important, Winfrey defined these tiles to live within so-called temperature N, temperature X systems. And he says if we have a temperature 2 system, for instance, that means that all connections which have 2 or more glues connected to each other are stable connections, and all which are less than 2 are unstable connections. Yeah? This is like in nature, consider Brownian motions. So if two molecules which fit together, which match, come together, then it's more likely that they stay together if they are not moving too much around, and it's more likely that they break up again if it's very warm and there's a lot of movement. Yeah? So that's the idea of this system, and let's check this. So we have two tiles here, and these sides here are the same, C and C, and what happens is that these two could come together and form a new structure. So that's the idea. The biological equivalent of this is DNA tiles, DNA strands. So we all know these DNA strands, the double helix thing. Here we only take always one half of this double string, DNA strand here and here and here. And these are the amino bases here, which we all know, at least have heard of. Let's put it like that. From these amino bases, we are able to create such structures here. And again, this is nothing special anymore. Yeah? So there's lots of commercial companies where you can ask for, could you please create me a structure like that? And then a week later you get, I don't know in what you get it, but you get these DNA structures and can use them for whatever you like. Yeah? So nothing special. And the question now is how do these map to the tiles here before to such a thing? How does it map? Quite simple. The interesting thing are the glues. Yeah, the glues are the stuff that is used for connecting the tiles, and you can more or less very easily construct very different glues by just creating these so-called sticky ends here of these structures here. So if you add, let's say, a few more of these bases here, then you can define very clearly what other kinds of DNA tiles might be able to connect to these. Because in DNA, always A and T connect to each other and G and C connect to each other. And if you have a C here, then you will need a G here. And if you have an A here, then you need a T here. So if you have another tile which has a sticky end with G and T on this side, then these at least it's possible that they connect to each other. That's the biological idea. And well, longer open ends result in stronger bindings. Yeah? So if you have 5 or 6 or 7 of these bases here, then it's much more likely that a connection that has been established, that it remains stable. If you just have one, then it's not a very stable connection. Yeah? That's the idea of mapping the glue strength here. Okay, and that's well, let's say an even more biological visualization with the DNA strands here and the sticky ends. Okay, so what can we do with that now? That's the basics so far. Old technology, more or less. What could we use that for? Well, we can use these tiles as basic building blocks for everything you need for a communication system within the human body. You can use it to build nano devices, you can use it to compute things, and you can use it to communicate between nano devices. And I suggest that we have a look at how this works. The first thing is, and this is nothing we've been working on, this is again stuff that has been done by Winfrey and others before, not always with DNA tile technology. There's another competing technology which is called DNA origami. You can probably imagine what this means. Yeah? So you have very long DNA strands, and by applying certain proteins at certain positions of the DNA strand, you can make them fold and then create certain structures out of that. Yeah? And already quite advanced technology, DNA origami. Actually, as far as I understood, I visited some guys in Harvard last year, and they said we prefer DNA origami very much over DNA tile technology, but just as a anyway, there's a lot of structures which have already been built, like the typical smiley stuff here, but also things like this, and this we'll use in one of the next few slides. So this is stuff which has already been done, which we are just using. Yeah, we don't work on building structures out of that. What we are much more interested in is communication and computation. Yeah? What I'll show you now is how we can compute logical formulas with DNA tiles. And actually, already Winfrey or someone else, at least already 15 years ago, proved that that the DNA tile system is Turing complete. You can do everything with that what you can do with other computers. Yeah? So let's take a simple example. Logical formulas, a four-way end. Yeah? So our DNA tile structure that we present now is, as soon as it is completely assembled, we know that the four-way end is true. Yeah? So then we evaluated the logical formula and found out that it's true. Yeah? When we do that. And how does it work? Well, first we need a certain, we call that a tile set, a set of tiles. And obviously, the tile set here needs some, let's say, some kind of some thinking of how we construct this the tiles here in order that they fulfill a certain purpose. But we have a lot of flexibility and a lot of room to play with that because we can completely freely define the number of glues here, and we can use labels here. On the mathematical level, and then we have to translate this into the biological level, but first on the mathematical level. And if you look at that now, there's in this model here, there's a starting tile. This one is so-called seed tile. Without that seed tile, the whole process wouldn't work, wouldn't start. But if we start with the seed tile, then there's not many tiles which can connect to this one here. Yeah, so it's not like Lego where you can attach every tile to every Lego piece to every other one. It's really like the puzzle because there only the matching. There's only one matching tile, at least should be, in high quality puzzles that you can use here. It's the same here. You touch the first tile here, that would be this one here. That's the only one which matches there. This is the only one which matches there, and then we can continue. I quickly go through this, but if you look at the sides here, it's always the same. Yeah? Here we have 2 and 2 and A, so these match, and because this is a temperature 2 system, connection with with 2 glues here is a stable connection. It's very likely that it stays like that, and so on. So I continue with that, and you see the red tiles here. These are the different atoms of the formula. Yeah, so that's A, B, C, D. And if all the 4 here are available in your tile set, then we can build the structure here. And these are again two special tiles. These are receptor tiles. And you see, if you look at the overall structure here, these are the only ones which now have glues to the outside. All the others don't have anything anymore. So there's no way for anything else to attach here, only here and here. So what we say is, this formula is true, or the formula that we constructed here is true if the 2 receptor tiles are available. And what we have to make sure is, at least to some extent, what we have to make sure is that there's an overwhelming probability that if the 2 receptor tiles here and here are in this position, then the rest of the structure must have been assembled like it looks here. Yeah? So in biology, that's not, it's not guaranteed. I mean, in computer science, we probably could guarantee that. Somehow in biology, you could not. It could happen that, let's say, this and this and this, that they connect to each other, yeah? Without all the rest here. And then some detector device which connects to these 2 glues here would say, okay, well, everything worked out nicely. We have the receptor here, the receptor here, everything okay. Yeah? So what we have to make sure with our system is that this doesn't happen, at least doesn't happen too often. Let's put it that way, and that these 2 tiles here are only at this position here when the rest of the structure has been constructed. So far so nice. Yeah, we have created a tile set, and when we bring this tile set together, then we just have to I'll talk about this in a second. We just have to wait a little, and then after a while this structure here would have been completed. Why wait a little? That's the idea of the let's say the mathematical model. What happens here is that you start with the seed tile here, and then you test the positions at the edge of the seed tile. Yeah? You test this and this and this position, and try all the tiles in this position. And once there's a match, then the tile will remain in this position, and it's the likelihood that it will be removed again is very low. And so, step after step, the overall structure here will be created. And this is also the way as it works in biology. Biology also tries the connection, yeah, because if you imagine that you put all these DNA tiles, so not these tiles here, but the biological equivalent, if you put them in a petri dish and just wait for a while, you stir a little and wait, then the likelihood that there will be at some point a certain connection coming up because the 2 molecules have met is rather high. Yeah? So this also works in biological settings. I said this already. And now comes the interesting part. Yeah? So this is more or less the basic idea of how we assemble these devices. But how could we use this in a medical scenario? Yeah? So if you just take one slide home with you, this is the one, yeah, that you should remember. We call that in-message computation because what do we do here? Let's set up our scenario. We are now in a, let's say, in a petri dish or in the human body or in a pig body or whatever. It doesn't matter. Let's say we are in a petri dish, and now we have a number of nanosensors. I'll say something about that a little later. So let's assume we have a nanosensor, something which is able to detect the presence of certain biomarkers. Now, coming to slides produced by PhD students, my PhD students tend to produce graphics in ticks, LaTeX ticks. which is a nightmare for me. So I was unable to recolor these. I would have loved to be some of them orange and blue and yellow and green. Yeah,
Text or animations. It's hard to tell, right?
And this would have been a nice topic for the discussion afterwards. Anyway, so these nanosensors, all four nanosensors are able to detect different biomarkers. Yeah? So let's say this is orange, these are blue, these are green, and this is red here, the biomarker, whatever that means. Yeah? Let's assume there's an illness which is indicated by the presence of 4 different biomarkers, protein combinations, whatever. And then these nanosensors would detect these biomarkers, and what do they do afterwards? So they've all detected these. They spit out more or less. They release these tiles here into the medium. Let's say to the petri dish. The petri dish already is completely filled with the other TB, the C tile, and so on. And what will then happen is that these tiles meet in the petri dish and more or less start forming this structure. Yeah? In the end, hopefully several of these structures have been produced. And what did we do? We computed the formula that these 4 tiles here are present. In other words, that the nanosensors here detected these biomarkers over there. Yeah? So the presence of these tiles here means the patient has illness X. Yeah? So what do we do with this information? We have to somehow communicate this. So for instance, there could be some other kind of nanobot. The message structure here connects to the nanobot and then releases some other kind of message. This could be, let's say, an IP packet, for instance. This could be a gateway device, maybe attached to the body somewhere, which is able to communicate in our normal, well-known wireless LAN way or something. Yeah? Let's assume this is an IP packet announcing, well, we computed this formula here, and there's a certain likelihood that the patient has this and this illness. And that's more or less the idea of this approach. There's certainly a number of things that this is the basics, and there's certainly a number of things that you can discuss about and what we've been working on, like security, like more complex protocols, and so on. So lots of things that you can work on here. But let's say the principle is, I think it's. I don't want to make this too long. I think the principle should be clear how this works. Talking about the nanosensors, you've seen one of these boxes here, yeah? the nano boxes on the slide with all these devices. This one here, actually this already has also been shown that this works. So we have this DNA origami technology here. And what you see here, this is a lid, this one here, which is closed due to the presence of certain proteins here. But if you attach certain other proteins, DNA markers, whatever, to the lid at a certain position, or if they get attached because they are present, then this will lead to, let's say, some kind of opening, because this and the two here try to have as much distance from each other as possible. And then the lid opens, and for instance, if this is filled with our DNA tiles, then they could get up. And this is also not fantasy. This has already been built. Nanoboxes like that. So this is a microscopic image of such nanoboxes. So also something which is already available and working. So again, no science fiction anymore. This is it's coming closer and closer to becoming reality. Okay. I'll shorten that. What we've done, and what is a very nice tool, is we build a simulator for these DNA tiles, for building structures, and especially for creating tile sets. As you can imagine, it's not, I mean, we are all used to programming in C++ or in Java or whatever high level languages, creating the tile set here is more or less programming the DNA nano network, is something we are not used to. Yeah? It would be a steep learning curve to learn how to produce these tile sets here. So what we are currently doing is we are creating a compiler where you have some high-level language which then automatically produces tile sets for you, which you can then send to whatever company currently produces DNA structures for you, and then they'll send it back to you and you can use that. But the simulator is quite nice, so if you want to have a look at it, NetTAS, quite easy to use. Output the tile set generator, I talked about this already. Due to the time, I'll skip most of the rest now.
As long as people don't flee from the session, feel free to.
Take your time.
Okay.
Okay, then let's have a look at gateway technology. So this is something we have not yet been working on, but which is rather, let's say, I would consider it rather simple to be adapted to our DNA tile technology. So what do we need? We need the connector between the inside and the outside network. Yeah? And so if you have a situation like this here, so there's some kind of tumor, and the biomarkers here have been detected, then we need to send the information to the outside of the network, at least in many cases. Sometimes it might be possible that the nano network already acts by itself, detect the tumor, kill it some way, yeah? But maybe that's not the usual case. Maybe the most important thing is tell it to the outside that something is wrong here. So there has to be a gateway, and then some guy here, let's say a physician, has to instruct the gateway to give a command to do whatever is necessary in the blood vessels and so on. So the question now is, how could gateways look like? How would this technology look like that you can send from the inside to the outside and the other way around information? And there's the gateway engineering people, they already came up with several ideas. So the first idea here is an engineered device with an RF and a molecular interface. So that could be an implanted device like this here. But it could also be, let's say, a nanoscale device which is operating within the blood cell, like this one here, and which is then able to signal the information somehow to the outside again with radio frequency technology. Or you might even have nanoscale devices, rather passive things where you can, where you have some kind of readable properties. So if there's, for instance, these little devices or particles available here, then you can detect it from the outside and also get some kind of information from the inside to the outside. And I just wanted to have a look at two examples. So this one here. So this is again a gateway technology which has already been built. So this is a field effect transistor biosensor which is basically able to let's say to catch certain molecules here in the inside, and once this has happened, and there have been quite a few of these molecules being caught by this device here, then the resistance will be different, and it will be detectable in that way. So something that works and has already been. Okay. Let's have a brief look at web lab and in vivo experiments. So this is something what we are more or less very close before doing such web lab experiments. So what do we think? How will this happen? How will this work? Well, we have to come up with a certain idea with an experiment. Let's say we want to detect certain biomarkers. Then we have to come up with a formula or with let's say a program that this nano network should execute or should find out. And then we have to design the tile set, hopefully with our tile set compiler. that the tiles be built by some company. So we give this plan, this building plan to the company, get the stuff back, pour everything into our petri dish, add water, stir, and wait until something happens. For instance, what's very popular at the moment in this community is fluorescence markers. So if you, so what my dream more or less is, we do exactly this, and then wait until the desired outcome occurs, that suddenly the petri dish starts glowing because this function has been computed. That would be, for me, that would be perfect. And this is the proof of concept, more or less. Our friends from our hospital and from the Lübeck hospital and from Charité with Falco Dressler together, they say, well, yes, nice, but if you want to win the Medical Nobel Prize, then you have to do a bit more. And they say at some point you will have to start with animal experimentation in order to see if this will work in the human body. Yeah? So in vivo experiments sounds nicer than animal experiments, but obviously at some point you have to do that in order to prove whether it works or not. So I think a nice concept, and still the question is, do we believe that this will work or not? What could be the problems that it doesn't work? And certainly two major problems are DNA or DNA structures might be destroyed by the immune system immediately, or the other way around, the immune system may go berserk because it's so overactivated, like with the corona stuff for several people, and things like that simply should not happen, must not happen, to be precise. This thing here, and this is technically interesting, this is important. This is a no-go, let's put it that way. So if something like this happens, then we simply can't use that. This is technically interesting, and we have a few colleagues at Charité, and they say, well, it's possible to create DNA structures in a way that the immune system simply doesn't see them. And you can even design DNA structures in a way that they survive a certain time X within the body. So you can say, I want to let this survive a minute or a week or a month. Amazing what is possible, but this is certainly very helpful. So if you want, let's say, if you want to create a monitoring network, then it would be very helpful if the network survives at least for a week or so. If you just want to find out in the next 5 minutes if there's something currently going on in the body, like a inflammation or something, then it's much less important to have long-living devices. So these two are important, and this one here is certainly also important. Gluing of tiles might work much worse in reality than in simulations or in petri dish. So in simulations it works very nicely, and I can very well imagine that in a petri dish this also works very nicely. So if you have let's say a restricted area or volume and you put in lots of these tiles here, then I would say it's very likely that at some point there will be connections between the tiles. But what about the human body? So does anyone know how long the blood vessels in the human body are altogether? 6 kilometers. So it will be rather unlikely that if you inject just 10 tiles of each type, that they meet. Right, and then you have to shake the guy. So obviously there's much more. We think it's much more unlikely, much more difficult in a real body. But anyway, there's again also people who already have solutions for that. Like you can try to, let's say, try to do some kind of compartmentalization. Is this the right word? So you try to keep the stuff together, and so let it travel together through the body. That could be a solution, but it's probably difficult anyway. Okay. Just a few words on medical applications, but not very much. So what we think is what we could do with that is targeted drug delivery with these nano devices. So what we have to achieve is once we have brought these nano boxes that you've seen to a certain position, then it would be very good if you could open them at exactly the position where they are needed. Actually, we just last year published a paper where we showed that with this technology we can localize the position of nano devices quite, not really exactly, but quite well, and then make the device open at this point. So this would be certainly something very helpful if you could realize this in practice. Then the stuff that I said that medical people think is extremely important, detection of very small quantities. That's exactly, by the way, the thing that the Harvard people say, and that's the group which is very much specialized in using DNA origami. So what they have developed is a system. Once they have detected a certain biomarker or a certain, let's say, set of biomarkers, then they also assemble certain DNA structures, DNA origami structures, roughly in the way that we do, just with the DNA origami, but in a way that exponentially creates more and more of these structures. So because these origami structures, once they have reached a certain size and structure, they split up in two, and these two do the same thing again as long as there's building material for that. Yeah? So they again increase in size, and once they have reached a certain size and structure, they again split up in two. And this makes it much more likely that this signal is detected. Yeah? So if you have an exponential growth of message molecules, just because you found 1 or 2 or 7 biomarkers, then it's much more likely to detect this. And this is obviously, that's a real advantage compared to our technology, which is not exponential. We just have our building material, and once it's used up, it's gone. Detection of complex diseases, yeah, so several professors all standing around this guy here having no clue what the problem is. With our DNA system, it's well, very simple to check for lots of different combinations of biomarker, combinations which might lead to problems. Okay, let's do that, and so far then I'm more or less done. So just to summarize, I think we have you have probably seen that well, Internet of Bio-Nano-Things is something which is not extremely far from reality anymore. We have come very close to really having all the technologies at hand that you need for building such a system. The question still certainly remains: does it make sense, and are there ethical and regulatory and so on concerns? Definitely, without a question. And one of our partners in our university hospital in Lübeck says, well, whatever you do, it will take at least 20 years until everything goes through to clinical trials and so on. Yeah? So no worries for most of us. This won't be a problem anymore. But yeah, it's technically technology level, or in terms of technology, we are really very far, and we are not so far away from realizing the whole thing. Okay, that's
Das ist ein Klassiker.
Okay, so my question is, thank you very much for the very interesting presentation. So my question is, can you speak more to the communications aspect? Because the biology in the body is that you have biochemical communications all the time, right? Yeah. So I was expecting more also that you kind of plug into that communication. Wouldn't it make sense to actually get the communication from that biochemical pathways that are happening and then act on it? That was completely left out. I was wondering why.
But actually, we would rather say that we want to keep out of the pathways as much as possible, not to disturb them, not to change anything in the pathways. So
you're assuming that read only is not possible?
Oh, you mean, okay. You mean just for getting information out of the
Yeah,
hardware?
and also then piggybacking on that communication, because somehow you have this assumption that the body is not able to do all of these things, but in reality the body is doing all of these things.
Yeah.
Maybe you might argue that, okay, it's not fast enough, not directed enough, and all of these things.
Yeah, but I mean, what I showed here does not speak against that. Yeah, so when I say biomarkers, you could also, let's say there's lots of functions that we've already modeled, like for instance thresholds of a certain amount of molecules that you detect,
Yeah.
and you can probably also do that, let's say, with white blood cells or so, find out if there are too many of them and things like that.
But this you also have from a normal blood test.
Yeah, which you would have to do.
Okay, but
So
doing
this-
a blood test is less invasive than.
Yeah, less invasive maybe, but maybe not as. Well, if you have a nano network which does monitoring all the time, then you can get the information much earlier than with a blood test. Because before you start thinking about, well, could there be any problem, monitoring nano network would probably be able to detect that much earlier.
My second question is regarding the DNA. I mean, can you speak more to? I mean, my impression was that not everything is reflected in the DNA. So actually, also manipulating the DNA is for very small percent. I mean, maybe the most severe ones, but a small percentage of illnesses. So not everything is DNA relevant.
No, but that's not what we are doing here. DNA for us is just building material. We don't use the information capability of DNA. We just use it as building material because it's very useful for that. Yeah. And actually we had this discussion last time when I presented this, and one guy said, well, why don't you make use much more of the information coding that you could do in DNA? Well,
That's
yeah,
a different
it's a
scope, basically.
Yeah, it's a different way of looking at it. So for us, it's just building material, and because you can design it so nicely, it's easy to create very different tiles and very many of them.
Because it's biocompatible.
Yeah,
That's why.
yeah, exactly.
Right?
Yeah.
Heraklit.
So your model is two-dimensional,
Ja.
Yes.
and it is square. So is it important, or could it be also three-dimensional?
This model is two-dimensional, but there's also a 3D tile model.
And to take 6 squares, why do you take just squares and not say triangles or 6 angles?
Well, that's the or hexagons.
Yeah, hexagon. Sorry, yes.
Or circles.
No. No. I think it should. How to say? It should completely cover the surface. So, but you can do it with hexagons and with triangles too.
Yeah, but well, I don't think this is needed. I mean, covering surface. What would be the advantage of triangles?
I don't know. Of course, that's a question to you.
I don't like triangles. No, but I mean, we basically use the existing DNA tile technology, which, as I said, was invented 20 years ago, and this is just established technology.
I would have expected you say because biology forces us to do it this
No.
way.
Biology doesn't work in squares.
No.
Yeah, that was.
No, but this is Winfrey, as you said. He came up with this abstraction. That's
Yeah.
it.
Or it may be one dimension only on the long sequences.
But you can use a triangle and build any kind of structure out of it.
Yeah.
Yeah.
Well, if you can do triangles, you can do squares. So
Exactly.
just think about using just 2 triangles together, if this makes it more conforming.
Das ist schwer.
Can't use that.
I mean, but in terms of, let's say,
Lego.
That's right.
Yeah.
available knowledge, there's a whole body of theory behind DNA tiles in this squared way, and it makes a lot of sense to use that. Maybe it's maybe it makes sense to think about, let's say, about triangles due to the 3D structures and so on. But for the moment, there's so much theory available with DNA tiles as they are at the moment. It's just easy to use them.
Thank you for the talk and thank you for the introduction to the computation of the DNA tiles. And since it's the first time I heard it, I wanted to ask, is there some way of automatically checking for unwanted combination of the tiles? Because I don't know, could it happen that they're combining in some way that might be malicious to the communication?
Absolutely. And what you have to do is you have to design the tile set very carefully in a way that the probability for this is very small. Okay. So, but it's simply in nature this will happen. When we design the tile set in a very careful way or very carefully, then we can make sure that the probability that, let's say, two tiles or more tiles which somehow got together, that they break apart again is very high. We have to achieve this. So you can't avoid that tiles which do not match stick together for a short term, and there might be a certain likelihood that even these two receptor tiles, that they are exactly in the position that they, let's say, can be detected in this position by a gateway or so. We can't avoid that. We just have to make sure that the probability is very small.
But there's like theory that says if I do this and that, or if I put this and that in my tile set, then the probability will be very small.
Yeah.
Yeah.
Exactly.
Thank you.
Have you ever encountered discussions about transhumanism in this context?
No.
Discussed it.
If there's no further question between Stefan and the beach and you, I would say we conclude the session. Thank you very much. It was as inspiring as I hoped for. So. Any news from the organizer?